Thanks, Kevin. Thanks for the introduction and the invitation to come over. I'm really happy to be here because complex systems are fascinating. This is something I didn't really realize a few years ago, so I'm relatively new to thinking about complexity, and I've come at this problem more from a biological perspective. So my group has been interested in a long time in building tools that help us understand function in the cell, and the complexities of understanding cellular function have caused us to think a little bit more about what makes biology and biological systems different from man-made machines and systems, if you like. And so I'll share some perspectives that kind of frame how we've thought about the problem, and then give a couple of examples of our own work that's taking a systems approach to cellular function and really open an invitation to talk to some of you more, because we found that we lack the analysis tools that I think are going to be important for driving our work forward. So I'm probably the most recent newcomer to complex systems here. So take all this as the kind of perspective of somebody who's more excited rather than knowledgeable about the area. So to get started, when one thinks about a complex system that is man-made, one recognizes that there are design rules and blueprints, and I mean, this stuff really makes sense. There are design rules that let us understand how systems are put together. If they break, we turn to the blueprints to understand how we can fix them. Oftentimes from the blueprints, we can predict how they're going to fail and when they fail. And when we want to go back later and augment the function, have them do something that they weren't initially intended to do, again, we can turn to the blueprints and understand how to augment their functions. And it turns out that if you look at a biological system, and the building block, I think, would be the cell, you find that we really don't have good blueprints, and as a consequence, we're not very good at intervening in these kinds of ways I've pointed out. So there was a period about 15 years ago when the human genome was being sequenced, and there was a race to see who would sequence it first, and then we had a sequence. The DNA in mammals is about 3 billion base pairs long. We got that sequence of A's, G's, C's, and T's, and we thought this would lead to a real turning point in our ability to treat disease, diagnose disease, etc. But it really hasn't. So if you think about drug development, this is the object of coming up with small molecules that bind to an enzyme and turn its activity on or off. This is still empirical. The way drug screening starts is Pfizer takes 2 or 3 million small molecules in their library and then tests them one by one to see which of those is active in blocking an enzyme's function. And then after those hits are identified, more empirical chemistry goes on so that analogs of those hits are made to just try to keep finding molecules that are a little better. So we're not able to rationally design drugs yet. If you think about diagnosing disease, we're still pretty bad at that. And that's something, in fact, that we all know. I mean, you go to the doctor, you've got something that's not right, they diagnose it, you get treated, it doesn't help, so you go back, they re-diagnose it, and this is a kind of an iterative procedure. But what's more interesting is that the clinical diagnostic tests that are done that look for basis of disease. We think of biomarkers, the molecules, the proteins that would be tested for to see disease. And a common one is the prostate-specific antigen, a protein that's detected in blood, which correlates, or at least increases in its level, correlate with the onset of prostate cancer. The FDA only approves 1 or 2 biomarkers a year. That means there are only a couple of new diagnostic tests approved each year. There's a need for hundreds of these tests. Work is going on to develop diagnostics for hundreds of conditions, but the pace is just incredible. The amount of effort that goes in and the success rate tells us again that we're not very good at developing diagnostic tests. And then predicting failures. We have a genome. We have of order 50,000 genes that are coded for in our DNA. And one question is, if a gene is defective, if the protein that's produced by a gene doesn't work, what's the consequence? Most diseases are caused by some genetic alteration of a protein activity. Well, we can't predict that. The way we figure it out is we take a mouse, we alter its DNA to stop expression of a gene, and we see what the phenotype is. We see what the mouse does. And so if we put human growth factor into a mouse, the mouse grows to be bigger. And that's a kind of clue that that protein drives growth in an organism. So that's still one of the tools we use to get at protein function and predict failure. So my point in going through these examples is to say that the kind of holy grail of figuring out the gene sequence of mammals and that giving us the instruction set, the blueprints for understanding biology, turned out not to be the case. Biology is more complicated and more complex, and there's more to do. So if you ask about what is the system we're trying to understand, I think most people would agree that the cell is the building block in biology. Cells come together to make tissues, which come together to make organisms. And cells are 50-ish micron particles. It's a lipid envelope that inside has DNA. I've told you it's about 3 billion base pairs in size. Of order 100,000 proteins can be there, about 1,000 small molecules, metabolites, things like ATP and glucose, and then salt, lipid, and water. So the parts list, if you think about it, today isn't so vast. We deal with systems that have that number of parts or components all the time. And the objective in trying to understand cell function is understanding how these molecules interact with one another inside the cell. Now, I want to show you a movie that was an animation by a Harvard team that really, if you haven't kind of looked into signaling and how functions operate in a cell, gives you a pretty realistic picture of what goes on, what the wiring in a cell looks like. And so I'll make some comments as this goes, but this here is cells that are rolling down a blood vessel. These are cells that are, this is more interesting now. So this is inside the cell. There's a kind of protein skeleton, an architecture that gives cells function and form. And that skeleton also serves as tracks that proteins move along, carry cargoes along, and organize themselves. A cell is spatiotemporally very structured, and that spatiotemporal structure is integral to cell function. And what else is neat is that these scaffolds, these protein scaffolds, aren't permanent. They're actually remodeled constantly. They're built in place where they're needed to perform a function, and then when that function's carried out, they disassemble to free up those building blocks that can then be harnessed somewhere else in the cell. And it's also the case that a lot of these proteins have multiple functions. So what function they're carrying out depends on their context, where they are, how they've been modified, and what their neighbors are. And so this is a motor protein that's carrying a cargo down one of these tracks. And this is cartoonish, but it really does a good job of giving you a two-scale picture of what those structures and dynamics really do look like in the cell. So this goes on. You can, actually I should have the reference here, it's not showing up, but there's a website that has this movie. I'm not playing the music. It's got beautiful music to it that makes it 10 times more stunning. And so if you want to go look at that later, you can. Okay, so let me go on and talk about a few other things that caught our attention. I think many of you think about this as well, of how a living device or computer is different from a solid state device. And one is the robustness, that biological machines fail gracefully, and we know that. If you are up late at night working on a paper or proposal and you've got to teach in the morning, you know, you get in there and you can teach and you're not 100%, but 85% is still passable. But you haven't gotten to that point where you ran out of energy and you just shut off, where a transistor broke and you stop functioning. We have this ability to fail and degrade gracefully. And I think an interesting example is if you take neurons in the brain, if you inactivate any 1% of the neurons, you can still perform most of the functions that you perform. If you inactivate 1% of the transistors in a billion transistor circuit chip, you've certainly killed that device from being able to do anything. And I think this idea that biological computers are built from imperfect components is interesting. When we design a chip, every transistor has very tight IV tolerances, and it has to. I mean, we have to know how those perform. If you look at neurons in the brain, they have a broad distribution of functionality, the potentials at which they trigger, their relaxation times, and this heterogeneity is kind of interesting because these are complex machines that are built out of parts that are, in some sense, pretty crappy. And so what are the design principles that let one build a robust system from imperfect parts? Obviously, biological systems are very adaptable. The notion that we can do abstract math, and it's not clear evolutionarily that that was selected for, at least not directly. But in a number of other ways, biological systems have this adaptation, not just through evolution, but a real adaptation. And then the kinds of problems biological systems can solve can be very different than the kinds of problems machines solve. And I think you know the one about identifying objects in a kind of a noisy environment, picking out Waldo, picking out a person in a scene. Biological computers, our brains, are really good at doing that. And machines have come a long way, but still can't compare to the ability of even simple animals to do that. And then biology is good at predicting the next step, at filling in missing data. And the group, my students, like this example, even though they're too young to really appreciate it. But if you watch that movie and it plays out, you know, I can stop it right there and you know what's going to happen next, right? Right. Computers are not very good at filling in kind of missing information and making predictions. And let's see if we're right or not. We are. You know, it's kind of funny. This movie was remade last year. I don't know if you've seen it, The Three Stooges. And took my girls to it. And the 9-year-old girl couldn't stop laughing the whole time. And I didn't think I liked it that much. So it's timeless. Anyhow, let's talk just a few more things about, no, not you. Let's go on. Good. So last kind of chart slide, and I'm going to get into a transition to what we actually do. But to make a few more observations, these living systems, neural systems, brains that I've talked a little bit about, and I showed you a movie about the cell, one can kind of ask, are there basic design rules, engineering principles that are used in biological function that don't have a strong analog in the way that we build human-made systems, okay? And there really are. I think this first point of being dynamically wired, it's really interesting. When we build systems ourselves and design them, components are wired up and that wiring is permanent. In biology, the connections between the components, which proteins interact with other proteins, that's dynamic. Some proteins a protein will interact with one set of proteins in the cell in one circumstance, but if that cell has been stimulated to cause it to differentiate, then that same protein has a different set of partners that it interacts with. And so there's this dynamic wiring that's contextual. And we're understanding how that's controlled, but it really means that there's not one static blueprint that shows us the connectivity of those systems, but that's constantly being manipulated, reconnected, and that's fundamentally a different way of thinking about the design. This kind of notion that, you know, a lot of biological systems don't compute to give one sole answer, but compute to give an answer that's good enough. The fight or flight response, the kind of pattern recognition response, emotional responses. It's hard to quantitate those in a digital analog, but that's very much how complex functions in biology work. Biology uses interactions that are highly nonlinear. That is to say, if you have a protein expressed at 5-fold higher level, the function that it leads to isn't increased by a factor of 5, but might be increased by a factor of 40. And so there are nonlinearities everywhere, And we know that by default, we like to design systems, man-made systems, that have no nonlinearities, that are perfectly linear. We try to engineer out, kill the nonlinearities. But a lot of the functionality in biology comes from these nonlinear relationships between component levels and function. And then this one, I think, is kind of among the most interesting, the spatiotemporal organization. A cell is by definition far from equilibrium. I mean, if it was at room temperature and everything was mixed uniformly, it would be at equilibrium. But a lot of energy is flowing through the cell. It's maintaining this spatiotemporal architecture, and that's intimately tied to function in ways that we still don't understand well and that biologists don't study well. So that kind of leads me into this idea that as we've learned more about the molecules in a cell, we've learned more that the ways that we study those aren't really appropriate for gaining deeper insight. So let me use a few slides here. Commonly, a biologist studies a signaling pathway. They've got a cell with a receptor at the surface. Some ligand binds to the receptor and stimulates a signaling pathway so that that receptor then might get phosphorylated on the inside, bind some proteins and brings a protein and an enzyme together. That leads to another reaction. Ultimately, a protein is modified so that it can get shuttled into the nucleus. And once in the nucleus, it can bind to DNA and turn on patterns of gene expression. And we think about this as a linear system. And biology kind of goes through the job of tracking through this linear series. So as this protein is discovered, we then try to identify what's it interact with. And then what's that interact with? How's it modified? Does it get into the nucleus? Where does it bind on DNA? But what you find is biology is messy. 3 different groups do the same basic experiment and don't get the same result. And we start to understand that these systems aren't so linear. They're not that's the wrong way to think about them, but there's all kinds of feedback, all kinds of contextuality. So that if you have a kind of linear sequence, there's a number of proteins that can feed in and modulate, alter the activities of proteins. And so you start to pay attention to more. And the more you look, the more you find that these systems are in fact quite interconnected. And these are the kinds of images that you all are much more familiar with than I am. I look at this and I say, this means that fly and worm and Clint Eastwood aren't actually that different. That has nothing to do with the empty chair. But the point here is that you look at these systems and you see a level of information and data that we in biology just don't know how to deal with, right? We just don't know how to deal with that. And this is kind of interesting. Somebody turned me on to this quote, this writing from a main philosopher a couple hundred years ago, that back then said every attempt to employ mathematical methods in the study of biological questions must be considered profoundly irrational and contrary to the spirit of biology. This notion that you go into physics or biology depending on whether you're into math. And you can, he didn't want to be misunderstood, so he went on and said, if mathematical analysis should ever hold a prominent place in biology, an aberration which is happily almost impossible, it would occasion a rapid and widespread degeneration of that science. Which, today some of us would argue is needed. So this really, I think, points to an inflection in the life sciences where biology is becoming a data science. It's becoming an information science. And we can continue to characterize the interactions of proteins in the cell, the activities of proteins. We can do it at higher kind of coverage at systems levels, and we can collect a lot of data. But data on its own isn't information or knowledge or understanding. And this is where biology is really running into a wall and creating a need, not just an opportunity, but a need to bring analysis tools in to help us understand what's going on. And that's exciting. It's also disruptive. It's disruptive because a lot of people won't make that transition, and so they're going to resist it. But that's always the case when new fields grow. And And I want to share a little bit of perspective that's driven our own work and how I see us as not solving the problem, but in fact trying to collect the kinds of data that will let us get deeper insight into cell function. So if you look at a systems level and you ask, how do we look at the cell in a systems level? Because I showed these linear pathways, I showed they get a little more complex, and if you look at everything, they're highly interconnected. The most important tool has been the gene chip array. So the idea here is that when genes are expressed, DNA gets copied into a messenger RNA molecule. That's then the template for making a protein. So if you have a messenger RNA strand present, that's basically a proxy for the protein being present. It's not perfect, but it's close. And so we can look for messenger RNAs by having a surface that has DNA strands on it that are complementary in sequence to the messenger RNA, so the message will hybridize or bind. Now that's kind of neat, because if your surface had an array of lots of different probes, then each, so this is a glass slide, each of these circles in today's technology is about a micron in size, so you can pack millions of distinct circles in the array. Each one has tied down to it a collection of DNA of some sequence. And so in one experiment, we can look for the presence of tens of thousands of messenger RNAs. And then that gives us a fingerprint of the gene expression profile. It tells us in that cell which genes are being expressed, which ones aren't. And that's an important part of kind of understanding cell function. But it's not the whole story. And this, by the way, is now, it's really incredible. 15 years ago, the first examples came out. They're still really expensive, thousands of dollars to do an experiment. Today, this technology has gotten to the point where an experiment costs a couple $100. There's core labs everywhere, and it's routine. It's so routine that graduate students doing it today can't imagine that a generation ago it was unthinkable to kind of have this data, access to that data. But gene expression isn't the full story. One also wants to be able to take this concept and move from DNA arrays to protein arrays and small molecule arrays, and be in a position to look at the protein activities themselves, because I told you that proteins have multiple functions, and their functions depend on context. So it's not enough to know that the protein is present. You also want to know what activity does the protein have. And proteins are themselves modified. They're chemically modified by phosphorylation, acetylation, and those are the kinds of inputs that control protein activity. So we really need to be able to see protein activity directly to understand the interactions. It's not enough to have gene expression data. So we got interested in this because the field about 10 years ago was trying to develop protein arrays and small molecule arrays, but wasn't having a lot of success. And I don't want this to become a chemistry talk, but I think it's neat to have some appreciation in different fields what the challenges are, what people are fighting against. So this is a good example. This was a paper in Nature not long ago of a small molecule array. Every spot on this glass slide had a different small molecule, synthetic molecule tied down to it. And the idea was that this group wanted to find a molecule that bound a protein of interest. So they fluorescently labeled the protein, they put it on the array, and if it bound, that should localize fluorescence to the spot. And so you can see the spot that this group pointed to as a discovery of a small molecule that bound that protein. But you can also see some other spots that are actually pretty good. And you also see that there's a high background. And you'd be right if you looked at that and said, that's not a really impressive experiment. That's pretty noisy. And the reason for this is that proteins absorb nonspecifically to materials. You know, that's how come medical devices foul, catheters have to be replaced after a couple of days. If you wear contact lenses, you enzymatically clean them. Basically cleaning proteins off materials. And this is really a surface chemistry problem. And my group, our kind of bread and butter has been surface chemistry for biological applications. So we were good at building surfaces that were biologically active, that prevented nonspecific protein adsorption, that let us tie down proteins in ways that maintain their function so you could still do assays. And to be able to do this so reliably, it could be scaled to arrays having large numbers of molecules. And the basic surface chemistry is pretty simple, as these things are once they're developed. We take a glass or metal plate, coat it with a thin layer of gold, and by thin I mean a couple nanometers. And sulfur atoms have a special affinity for gold, so if you treat that, you just immerse the plate in a solution of molecules that have a sulfur at the end, the sulfurs will attach. They'll attach to the point that you have a dense packed array. That causes these chains to be in an extended alignment or conformation. And the nice feature of that is that we can synthesize surfaces by using chains that are synthetically modified with different end groups. So if we want to synthesize a surface that has some molecule, we make the chains that have that molecule and then let those self-assemble into this monolayer, and that gives us a surface. So that actually works pretty well. So the kind of way we would do an assay, and I show a couple slides on this, is shown here. Kinases are enzymes that phosphorylate proteins. In human, there are about 500 kinases. So that's a big fraction, a percent of our genome codes for kinases. They're involved in every cellular activity, and most cancers underlying most cancers are kinases that are constantly on. So kinases have the ability to be turned on and off. Growth of cells requires kinase activity. When you turn the kinases off, the cells stop growing. When these kinases undergo mutation that stops their, that inactivates their ability, that's a bad way of saying it, undergo mutations that leads to them being constantly on, then you get uncontrolled cell growth. That's cancer. So a lot of drugs in cancer are designed to bind to these kinases and block their activity. So this is an assay. This is a peptide sequence that we tie down to the monolayer. And then if we treat that monolayer with the kinase, in this case Sarc, and ATP, the source of the phosphate, the enzyme will phosphorylate the peptide. And how do you see that? If you run this reaction, you need to be able to see that the phosphate group is present. And the normal way to do that is to use a radioactive group. P32 isotopes decay, so we can look at radioactivity. So if we run that reaction, rinse off the surface, we see radioactivity that tells us the phosphate group has been transferred. This is kind of interesting. These are control experiments that shouldn't give us any activity. And you say, okay, your surfaces aren't perfect, right? You're getting a little bit of background, except this is the control where we put in no P32. So it's impossible to have radioactivity. But this work was done at the University of Chicago, where I was a professor for a while. We were in the Jones lab. The Jones lab is actually the site of the Manhattan Project that isolated the first unstable element. And this is, my experiment that shows that it's still there. So that's one reason, Julio, I was happy to move. It's safer here, okay? And I like to say, 12 years of a PhD, that's a minimal exposure. So that's just a joke. Anyhow, so if you subtract that out, our signal to noise in this assay is like 80 to 1, our dynamic range. And that's stunning. A lot of the assays that are used in biology, Western blots, ELISAs, working ranges of 6, 7, 8 to 1 are considered pretty good. And so to have this kind of range, low background, makes a big difference. And we show that you could do quantitative assays. A lot of times people use these chips, these solid phase assays, and regard them as qualitative screening tools. But that's just because they didn't perform well. If you take care of the surface chemistry, you could look at activities that can be modeled, that are good data, and do everything on the chip that we would normally do in kind of more conventional experiments or assays. So that's where we were. And we were really excited about extending this to look at other classes of enzymes, enzymes that perform different kinds of reactions. And the hard thing, the frustrating thing in all of this is that you have to be able to see the product of a reaction. You have to label it. In this case, we label that reaction by using a radioisotope. Sometimes you use an antibody that binds to the product of a reaction, and the antibody is fluorescently labeled. There are different ways to label, but what it means is new assays take a lot of development time. Some activities are hard to label. There's not a natural way to work a label in. And you can't run an experiment looking at different activities simultaneously because the labeling protocol for one activity is inconsistent or incompatible with the labeling protocol for another. So that led a number of groups to look for what we call label-free tools. Can you look at these activities without labels? Can you see them directly? A lot of the work used optical methods, evanescent wave technology, basically looking at electromagnetic properties of molecules near a surface. They don't give you a lot of information. We had found that mass spectrometry worked really well. And this is actually remarkable that it wasn't done sooner. This was about a decade ago. If you put these monolayers in a what we call a Maldi-Toff mass spectrometer, you pull it down in vacuum, hit the surface with a laser, that energy leads to a kind of a desorption of the molecules into the gas phase, and then they're accelerated in a field to a detector. And their time of flight is a function of their mass and their charge, and so you can figure out the mass. For us, the amazing thing we found was these monolayers desorb by cleavage of the thiol gold bond, but the molecules themselves don't get fragmented. And so they fly intact to the detector. And that gives us really simple spectra. So if I do that kinase assay again, we've got a surface that has the peptide on it. If we immobilize a peptide and take a mass spectrum, you see that there's mainly one peak. And it's at a mass of 2100, which is consistent with the mass of the peptide alkane thiol conjugate. And that little peak to the right, that in fact is the same species. The first one is a hydrogen H+ adduct. The other peak is a sodium ion adduct. So it's just a different counterion. We then treat with enzyme. We phosphorylate, and you take a spectrum, and you see that the mass is shifted by 80, and that's what it should do if you phosphorylate. So this was great because now we see every molecule that's on the surface, because every molecule has a mass. And if we do a reaction that leads to a change in the mass, we see that product. And so this format is label-free, and it can look at all activities. So this is another one that I'll come back to. This is a peptide which is acetylated. This is one of those chemical modifications that controls function. If we immobilize it, we see the species at 1990 mass. If we treat with an enzyme that deacetylates, then the mass comes down and we see the product. So we can follow that activity. And we did this, in fact, with a whole series. For a period of 5 years, we were applying this to different kinds of assays and activities. And it worked well. And it should work well because, again, we're just looking at changes in mass that follow reactions. So this gives us a pretty general way then to create arrays, treat those with biological samples, and then be able to read the activities. And I'm going to just tell you one thing in passing here that is really neat. So if you make an array and you do an experiment, you're looking for fluorescent signals that tell you there's activity. And one problem with these arrays today is the quality control. You may think that there's a protein in one spot, but in fact it never got immobilized, so that spot is blank. So that can give you a false negative result, right? But the other thing is, when you're using labels, you're only going to see the activity that you're looking for because the label is revealing one kind of reaction, not others. Which means you can't discover new reactions because you're looking for the keys under the light pole. With label-free methods, it's possible to take a sample, treat an array, and actually see a change in mass that you don't understand what that's due to because that reaction hasn't been understood before. So with these label-free methods, not only can you look at a collection of activities in one experiment, but you can discover new modifications. And I'm not talking about that today, but we've had some success with that kind of thing. One kind of fun thing is when we were doing this experiment, this kind of work, we still weren't doing things at high throughput. We're still doing things manually with a pipetter, running 50 reactions at a time or so. And then we had a couple guys in the group, Mike Scholey and Adam Eisenberg, want to automate this. So automation means working with plates that are about that size. It's an industry standard in the life sciences. And they have 384 or 1536 or factors of 4 greater spots. And these guys work this all out to be compatible with robotics so that we can have robots run thousands or tens of thousands of reactions and then deliver those to the array plates, and molecules get immobilized to the surface, and then we analyze them. Each reaction now is a microliter in size. That's huge because a lot of times these experiments are sample limited, and if we can squeeze 100-fold more experiments out, that makes a difference. And then these plates also get read in an automated mode. So that instrument's a mass spectrometer. Whoa. Is a mass spectrometer. It accepts plates that go in it, and then they get read. So that's a spot that's being analyzed to give a mass spectrum. This is going back to the front row. It's giving a spectrum. It goes to the next spot. This is all automated, and it's about a second per sample now. So we can do 50, 60,000 samples per instrument in a day. So then we had the problem that we're getting more data that we could look at. One student can't look at 10,000 spectra. It's impossible. I mean, it would negate all of the benefit of doing things at high throughput. So then we had some software written by some contractors that basically give us now several customized tools for scanning through that data, pulling out the kind of relevant spectra. So what I want to do in the last 5 minutes is give you basically an example of the kind of work we're doing now and invite some discussion afterwards. And this is a really hot area in biology and drug development now. Some of you may have heard the term epigenetics. And this idea is that if you look at somebody's DNA alone, you don't understand all of their function, because DNA can produce proteins that can be modified in different ways to alter their function, and that can be heritable. A daughter cell in a dividing cell can get those same modifications to proteins. So in some sense, a cell can inherit information which is not coded for by DNA, but which is coded for by modifications of proteins. And that's the argument that looking at DNA sequence alone, having discovered this DNA sequence, doesn't give us all the information, because it's really at the protein level that functionality changes and leads to disease. So a kind of classic system now, it's still a new system where this happens, is in the nucleosome. So you know, if you take our DNA and you stretch it out, it's about 3 meters. And every cell has a full copy of DNA. So you're packing a long molecule into a 5-micron nucleus. And the way that's done is the DNA is wound up on these cores. They're tightly packed, and that gets more packed into a chromosome look, the X look. So that core is a protein aggregate. This is the DNA that's wound around it. And this protein aggregate has these kinds of tails hanging off. These are peptide sequences. And these sequences are sites for these chemical modifications. And this is really remarkable, because if you look at one of these tails, histone 3, this tells us where phosphorylations are known, where methylations are known, and where acetylations are known. And I mean, don't worry about the details. These are the amino acids, so lysine, arginine, alanine, etc. But the point is that if you understand which modifications exist, and then you ask how many combinations of those modifications are possible, it's several million for H3. So this protein, which is one protein, which is coded for by one gene, can in the cell exist in several million forms. So that complexity is striking. And we know that different modification patterns, different states if you like, correlate to different cellular activities. A cell which is differentiating, a stem cell that's differentiating into a neuron, will have different marks on it, different modifications, than does a stem cell that's differentiating into a hepatocyte, a liver cell. And what we don't know is what the redundancy in those states are, which ones occur, which ones don't occur, et cetera. Okay? And a lot of biology is, again, taking more of a correlational look. Our interest has been in finding, in trying to understand molecularly how a cell installs these marks, correlates those marks so that when one is present, another is present, and then modifies them. And the remarkable thing is that if you look at acetylation, there are only 40 enzymes in the cell that control acetylation state. That's not that complex. I told you earlier there are 500 kinases that control phosphorylation, plus 150 phosphatases. So this is 40 catalysts, 40 enzymes, kind of regulate acetylation throughout the cell. That is, they regulate acetylation on thousands of proteins, and we don't even begin to understand how that system is optimized to have that regulatory power. So what I'm going to do because of time is I'm just going to skip to this and talk about a kind of a gene chip experiment. So here's the idea. We now know, and this is very recent, 2009, a paper came out from a mass spectrometry group, a proteomics group in Switzerland, where they looked at the proteome in cells and they could identify proteins that were acetylated. Now before this work, we maybe knew there were 60, 70, 80 acetylated proteins in the cell. And these guys found that there were thousands of proteins, I think, well, this case, 750, that's grown. There are thousands of proteins that exist in acetylated forms. This is just a few years ago. 5 years ago, we didn't recognize that acetylation of proteins was a pervasive modification. So when we were thinking about biology, we weren't considering how acetylation could impact protein function. We're just totally ignoring it. And this work has shown us that acetylation is a regulatory modification. Proteins get acetylated, they get deacetylated, and this shifts their activities. So this is part of that dynamic wiring of interactions. And we still don't understand how acetylation patterns look in the cell, how they're changed, how they vary in different cells. And so we got excited about this because we had a good tool for looking at acetylation. So the idea is, if we had a chip with different proteins on it, a panel of proteins, we could analyze to see which of those were acetylated. So the way we do the experiment is we grow cells up, or you take a biopsy tissue, you create a lysate. That's just a fancy way of saying you put the cells in a blender and you spin it. Okay? Then you spin it down in a centrifuge to get all of the particulates out and give a solution. That's a lysate. It's got the enzymes in it. So we apply that lysate to every different spot on our array, and if that lysate has a protein that will acetylate or deacetylate the protein on our array, there'll be a change in mass. And when we do the mass spec, we see that change. So for a given cell lysate, we can look at the levels of acetylation activity on a series of peptides. And the notion is that if you compare healthy cells to transformed or metastatic or cancer cells, there would be a different pattern of activity, and that would then lead to more detailed biology to try to understand why. So the neat thing we found is that we could use our arrays to look at these activities in lysates directly. So this is a kind of scaled down example, but here are a series of protein segments that were put on the array, and this experiment was done in mice livers. So we literally had mice growing, and the study had to do with how metabolism affects these activities. So some mice were on a high-fat diet, some were on a low-fat diet. And the mice were sacrificed, their livers were digested, and those liver extracts were put on the arrays. And there were two mice. One is wild type, it means a normal mouse, and one was a Sirt3 knockout. So this is a mouse that didn't have the gene for that protein, Sirtuin-3. And Sirtuin-3 is one of the deacetylases. So if you compare wild type in blue to knockout, what you see is this peptide shows a decrease in activity. So you would think that that's due to Sirtuin-3. If you lose Sirt3, you lose some activity. These peptides don't have any difference, so that Sirt3 must not be the enzyme that's involved in modifying this peptide. And you get patterns of activity. Now what's neat is we could actually make those proteins separately just using molecular biology. So if we make Sirt3 and test its specificity, we find that it agrees with what we see in this complex sample. So if you look in green up here, these two green spots are peptides. So this is a peptide sequence. X and Z are varied according to those amino acids and those amino acids. And these two peptides, the sequence is YKL. YKL is one of the peptides that has less activity in the knockout mouse. And we also see that that's a good substrate for the enzyme. This one in red is DKH. DKH has low activity, but it's not changed when we knock out Sirt3. And our array experiment shows that it's not active for Sirt3. So what we're doing is using panels of peptides and proteins as sets of substrates or probes, treating those with extracts and lysates, mapping out profiles of activity, and seeing how they change in different cells and disease cells, in cells that have been stimulated with drugs or other things. And it's working really well, so that we're able to again take complex samples, profile them, and get these data. We've kind of set up the software so that when these plates are scanned, all of the processing happens within a few minutes. These heat maps are then generated, and we're able to acquire data that's unprecedented. It's the first examples of looking at this deacetylase activity in real samples. And our problem now is we don't have good ways of analyzing them. Even though we have a lot of information, we know what the basis activities are. Out of these 40 enzymes, we can characterize each one independently to know what its specificity is. And then when we look at these activities in the cell, it's going to be some combination of those. And there's a lot more information in there. And we're at a stage like the early days in BioChip that we can do the experiment better than we can analyze the experiment. And so that's kind of my ending to the talk, that we're eager to talk to people who've got an interest in these kinds of systems, working with this kind of data. And there's a lot more there that I haven't shared, obviously, but we're kind of eager to engage with some partners that are excited about this kind of analysis. So that's the talk, okay? And the kinds of conclusions I'd make are a couple things. One is that in biology, you know, the mainstream biology is organized around hypotheses. When you write a grant to the NIH, the so-called R01, the main grant, you have to have a hypothesis and then you logically set out and test it. But what does drive biology? Is it hypotheses or is it tools? And if you look at the role the tools play, and again, some of you will be familiar with these, some won't, but in biology we use antibodies all the time. When we probe these signaling pathways, we do something called Western blots that all use antibodies. It's unthinkable to imagine what biology would look like if we didn't have antibodies. We use PCR to amplify DNA. Unthinkable to think, to envision biology without PCR. Green fluorescent protein, which won Roger Chen a Nobel Prize a few years ago, is a way for us to light up a protein in a cell. It's routine now. But without that, we'd have much less information about protein function in the cell. And the list goes on and on. Solid phase synthesis of DNA, oligonucleotides, protein synthesis, etc. So I would argue that, the real disruptions in biology stem from a new tool, not from a hypothesis. And I'm not saying that hypothesis-directed research doesn't play a role. It clearly does. But I'd say it's a more local role. When you basically know what you're working with and you want to know, does it do it this way or that way, you study it. Sometimes you get a surprise that you follow up on and it's a big deal. But if you come up with an enabling tool that cuts across all areas, that's the kind of thing that really drives the science forward. And these tools for looking at protein function in cells and tissue, I think represent what's still the early stage of a good example. So I'm going to stop there. Thanks again for coming out, and I'd be happy to have some discussion with anybody. Thank you.